So this is the second section, or the last section here, in 2.4 on mutations. This is looking at how we can trace genetic diseases. And this is like one of my favorite parts about genetics, besides Punnett squares, is pedigree charts. So pedigree charts are a genetic family tree that uses specific symbols to show the genealogy and occurrence of particular traits. Pedigrees can be used to predict the genotype of an individual. So if we can go to something that'll give us some examples, yes. So certain symbols in a pedigree chart, a circle means you're a female and a square means you're a male. If you're shaded in so that you look that like black circle versus a white circle there, shaded in makes you affected. So you have the disorder, disease, whatever it is that we're looking at, the gene, and not shaded would be someone who's not affected. And then if you're joined together, that means you are mated. So as far as this one, this means that this is like the mother. Why is this not working? This is the mother, this is the father, and then this would be the child. So when you see like someone coming out of a male and female, that's the child. Okay? So this is sort of like a scientific way of doing genetic trees, or family trees, I guess. So the first example of a family tree, or how we can use these pedigrees, is autosomal recessive. So by looking at this family tree, this pedigree chart, we can figure out just by looking at that that's an autosomal recessive gene. So if you go back in this lesson, you'll see that autosomal, the word autosomal, means that it is not linked to sex. So when something's autosomal, it means you can see it in males and females, and that's what we see here. It's in males and females. Something that's dominant should show up in every generation. And if we're looking at this one, we don't see anyone in this second generation that has the gene. So this tells us that it is autosomal recessive. So it might skip generations, making it recessive, and then it shows up in males and females, making it autosomal, okay? Is it possible that the pedigree below is for a recessive trait? Write the genotypes of each individual next to the symbol. So by looking at this one, the question's asking, is this a recessive trait? So in order to be recessive, this parent would have to be, let's use A's, little a, and little a, little a. So the only thing that this child could get would be little a, little a. So if it was a recessive thing, then this child should have it, but they're not shaded in. So let's show another example, and we can use Punnett squares too to help us understand this. Let's say that it is autosomal dominant. So let's say it's that. So if we go back to what we've learned before on pedigree charts, Punnett squares, we can do a cross between this. And if we do that, we will get this one, and this one would all have the trait if it's dominant, and this one wouldn't. So there is a chance that if it was a dominant gene, they could have a child that doesn't have it. Unlikely, but possible. So the question here is it possible for this to be for a recessive trait? The answer is no, must be dominant. Must be for a dominant trait. And the parents must be heterozygous. So they must be for a dominant trait, and the parents must be heterozygous. So heterozygous means that they have two different types of alleles. All right. The next one is it possible for this to be an autosomal recessive trait? So showing up in males and females, we can't tell because it only shows us one female that's affected. Typically, we know that if it's a sex-linked trait, then it would be showing up in males. It's not showing up in males, so we would assume that is autosomal. And then if it's recessive, let's see what a parent's might be. So if the parent was big A, big A, little a, then we did that on the last slide. There is actually a chance of the child being getting the recessive. So if this was recessive, autosomal, then that might be what this is, and then this child was one of the other ones, one of the other genotypes, making them not have the disease. So my best guess here is that yes, I'm not saying it's a guess, but from looking at the pedigree chart, an educated guess is that this is an autosomal recessive trait. Okay. So something that's autosomal dominant, remember that's something that is not related to sex, so can show up in males and females, and then it's dominant. So if it's there, it overpowers all the rest of the genes. It says, look at the following. Is it possible for the pedigree to be autosomal dominant? Didn't we just do this? So if it's autosomal dominant, if you have the dominant gene, that means that you have the disease. So these parents don't have the disease, they must be that. So if you were to cross someone who is little a with little a, all of your children are going to be little a's, which means that this child would never be able to have a big A or something to make it. So is it possible for this to be autosomal dominant? No, it has to be recessive. Is it possible for the pedigree to be autosomal dominant trait? We already talked about that. Okay. An extra practice. It says, is the following pedigree for autosomal recessive trait? Remember, this means that you must have two recessive traits to be affected by the disease. So again, what I've told you, autosomal means it shows up in males and females. We see that it shows up in males and females, so probably autosomal. Recessive means that it normally skips generations. Look, it skipped generations. So is this autosomal recessive? Probably. And if we really wanted to go even deeper, we could say, okay, if this is recessive, let's say that dad was big A, that was little a, all of the children would be that. This girl, she's not part of their family, so we actually have no idea her genetic background. These people didn't have kids. This guy has a kid with this girl, and we don't know what her genetic makeup is, but let's say it's big A. So let's say one of their children is big A. Not that one. This one gets that, and this one is this. So if this girl, their daughter, has a kid with, I don't know, let's say this guy who is big A, little a, then they could have children that could show up as being little a. Okay, so yeah, very possible that that is an autosomal recessive. So this is a little bit of a detective work sort of thing, and using the Punnett squares to figure out if it's possible for these pedigrees to be for certain traits. We've also talked about this already. The conclusions that you made for autosomal recessive traits apply to X-linked. In the exercise, you'll work on some additional conclusions because males have only one X chromosome and females have two. We will determine if the pedigrees below can be for a trait that is X-linked recessive. Use the following designations. So a normal X, we're going to use big A. The disease or the abnormality is going to be little a, and Y is for males only. So it says, write the genotype of each individual next to the symbol. Is it possible that the pedigree is for an X-linked recessive trait? So same applies. We can't see whether it's only males or females, so we don't know if it's X-linked or not, but we can probably get that it's recessive, likely, just because it's not showing up in this generation. So let's say this girl has little a. Oh yeah, we're doing X-linked, sorry. So she's got two X's. If she has the disease, let's say this guy doesn't, then their son will get an, yeah, that wouldn't work. So is it possible for this to be X-linked recessive? The answer is no. And the reason why is that if I did it like this, since the dad doesn't have the disease, he has a big A there, I guess. Little a, little a. If we cross this, we get this. And if we're looking at our child here, he would he's going to get his Y from his dad. That's how it works. And then his X is going to come from his mom. So regardless of what X he gets, if you look at these, they both have the disease. And he doesn't. He's not shaded in. So that would make this impossible for that to be an X-linked recessive, okay? So erase all that. We've got to start over. So it's not X-linked, but it could be autosomal. I think that's what I was. I was already starting on it being autosomal recessive at the very first. When you've done millions of these, you can see it right away, but I just wanted to show you that process. So if it is autosomal recessive, let's put her as little a's, and this guy as, I don't know, he can be big A or big A little a. Regardless, the child will get one little a from the mom and a big A from the dad, and then he will not have the disease. So autosomal recessive, that's what this would be. Here we've got to write the genotype for each individual. Is it possible for this to be an X-linked recessive trait? I'm going to take a wild guess and say no, but let's look into it. So we're checking if this is an X-linked recessive. So the mother has it, the father doesn't, and then the child has it. Well, I guess it could be X-linked recessive. Okay, so if we do that cross, well, we don't have to do the cross. We're always going to get a X little a from your mom because she has the disorder. And then we're going to get, because this is a male, a Y from the dad. So when we get that bad X from our mom, we have nothing to overpower it, so then we have the disorder. So the answer is, can this be an X-linked recessive? The answer is yes. So this would be X-linked recessive if the mother had the disorder, okay? So we always, when I said it's normally males, if a female isn't affected by it, then she could be part of the X-link. She's not just a carrier. So it's definitely possible that females could be affected, but once they're affected, we know exactly what their genotype is. And their genotype here is gonna be little a, right? This guy, he's affected, and then this guy, he's good. All right. So there's some practice problems on that on page 124. I'd like you to do 42 and 43. We're going to move on to finish off this section. So what are some beneficial things in the population or from mutations? Well, mutations introduce new alleles into a population, and I've already said that. If the new gene or the allele gives an organism a survival advantage, like I said, when you have webbed feet in a world where you're drowning, everyone else is drowning, you're a good swimmer, that gives you a survival advantage. You're gonna likely survive, and if that helps you to survive, you could possibly pass it on to the next population or generation. This will eventually change the characteristics of the population, and this is known as evolutionary change. A little picture of this, it says a gene produces an enzyme that makes black pigments to color on an insect's exterior. So all those little black dots on the leaf are black little beetles, I guess you could say. And then it says a mutation to this gene causes the enzyme to produce green pigment instead of black. So now some of them, most of them are still black, and then this one right here, it is a lighter color. And then it says the individual colored green blends better with the surroundings, so it's more difficult for predators to locate it. The green colored insects have a better chance of survival and therefore improved reproduction. So eventually, because all of these black beetles stand out, they're going to start to be less of them and more of these ones because they blend in and you can't see them as much, so they are less likely to be eaten and better chance that they'll survive to be able to pass on their genes. Okay, so that's an example of how mutations can be definitely a good thing and they can lead to evolutionary change. So sometimes people think it's hard to believe that we've evolved from, I don't know, fungus or something like that. But this didn't happen overnight. This happened over millions and millions of years. And these little small mutations that happened along the way have created us to be who we are today, and every other organism as well. So there's resistance in bacteria. Bacteria that have a mutation that gives them resistance to an antibiotic will be the bacteria to survive and reproduce. This is known as survival of the fittest. So this is when your doctor gives you antibiotics and you don't take them, or you don't take all of them. You're gonna introduce the antibiotic, and then your body is going to not be able to fight it off. So then the bacteria will get stronger, and then the next time you give them that antibiotic, they're going to be even stronger because you've produced bacteria that is now already knows what an antibiotic is, and it's going to be harder to kill it off. So that's an example of survival of the fittest. Here's a little picture of it. So it says your original bacteria colony, 95% of the bacteria are killed, 5% survive due to a mutation. The survivors grow and form a new colony. the survivors are resistant to both antibiotic 1 and 2. Then the survivors form a new colony and the process continues. So that's why we're always having new bacteria, we're always having new viruses. Well, not viruses, we're talking about bacteria here, but we have new forms of bacteria because they will evolve. And bacteria evolve much faster than us just because of their lifespan. So that in a very short period of time, we can see things that are already evolved. Our practice problems here are on page 128. We have question 1 to 5. I'd like you to do that, right? And then this last piece, I don't think it's really too important. Well, yeah, I'll show it to you. It's only a couple slides. So this is actually section 2.5, just connected in. And it's talking about genetic technology. So DNA fingerprinting and profiling is something that we use all the time now. It's mostly used in, well, the way that we think of it, it's used in like crimes and stuff like that. So finding DNA at the crime scene and matching it to someone else. That's very true. That does happen, as well as in like paternity tests. So if we're testing to see if someone is like your parent, the classic Maury, that is also how we use DNA fingerprinting. But anyway, so the steps to do DNA fingerprinting is that we take a sample of DNA. It's taken from a crime scene. We add enzymes that will cut the DNA, and then the DNA will cut. Cuts are added to a special DEL to create a pattern. Radioactive substances are added and are used to develop the fingerprints. That's a very simplified version of what actually happens. But basically, electricity is run through it, and they, due to their electrical charge, they'll move along this gel sort of in a certain pattern. And if I ran my DNA against my own DNA from a different sample, it should show up the same. Like I was saying, they also use this in paternity tests. So if you're a child, you should only have only bands that are from your parents, so one parent or the other. So if this is the child, and they're trying to say that these two were the parents, that's not possible because this band is not in either of the parents. Okay? So no, that's not the parents of that child. All of the other sets of parents have that band, so we would need to look. One that's really sticks out to me is this band. So if we look, this parent has that band. But I guess so does this parent. So maybe not. If we can look for some other bands, we got those two bands there, we don't got them. Yeah, so you're just trying to match up the bands and they have to be in one parent and the other. I'm pretty sure that this child goes with these parents, just from looking it over, but basically that's how you use DNA fingerprinting, so just lining up the pieces. Transgenics is a type of genetic modification in which the genes from one species are transferred into the DNA of another species. This is called genetic engineering and the modification of genetic material through actions of people, selective breeding, and modern techniques outside the normal way of reproduction. So transgenics, putting DNA from one organism inside the other, and then genetic engineering is basically just any sort of mating that happens that we have sort of like modified from like the natural way of reproducing in humans or animals or plants. We use transgenics and genetic engineering to create genetically modified organisms. Some examples, we are inserting some human genes into pigs in order to make the organs in pigs more compatible with human organs. so we can potentially use them for transplants. All right, so we're trying to create, I guess, organs inside of pigs. There's lots of other things that we do. We use transgenics. I know the classic one is tomatoes. They've put genes from fish into tomatoes just so that they can grow in colder environments. So yeah, many different genes from different species that are put into one another. And sometimes that leads to some allergies just because people are allergic to fish. But they don't eat fish, they eat the tomato, and the tomato has genes from a fish. And then all of a sudden they start to get a fish allergy, and they don't know why. Or sorry, a tomato allergy. So there's many different reasons, and I have actually noticed this I'm allergic to kiwi, and I've noticed this with different fruits. So possibly genetic modification of different fruits have caused there to be like kiwi genes in the fruit. I don't know, but I can find sometimes I'll get like a scratchy mouth after eating other fruit that I normally wouldn't affect me. So anyway, that is all of the biology unit. Finish the questions that go along with that, and next week we will begin and finish the physics unit. Okay, so let me know if you have any questions, and have a good weekend.